Okay, we're going to go ahead and get started with the afternoon session, Angiochem, and we look forward to hearing the work on blood-brain barrier transport that she's going to talk about. If Ralph De Libero was here, he would say we've been trying to get Angiochem to talk for a number of years, so we're glad to finally have you, Jean. Thank you.
Okay, thank you. And I'd like to thank the organizers for the opportunity to come and represent Angiochem here today at this excellent venue. A bit of a disclaimer, I am fairly new to the company, having joined this year, and I'll be discussing a lot of excellent science that Angiochem has done, in most cases over a year ago. So if I use the word we, I don't technically necessarily mean myself. So at Angiochem, we have a technology which is sometimes referred to as EPIC for engineered peptide compounds, which allows drugs to get through the brain using an LRP1 receptor mechanism. And that's what I'm going to describe for you today, using some various examples. This slide is just to orient everyone to the blood-brain barrier. I think everyone is familiar with the challenges that this barrier presents to drug discovery, from small molecules up through biologics. It's comprised of capillary endothelial cells, which are sealed together with tight junctions. There are also, as was mentioned in an earlier talk, efflux pumps, which push things out of the brain, especially things that are trying to get in. There are also astrocytotic feet lining these capillaries, and cells called pericytes. And this has been a major challenge to drug discovery. However, there are molecules that do need to get into the brain, such as nutrients and hormones. And those molecules have their own way in. And so we were thinking maybe we could use one of those ways in for drugs. So to describe that way in, I'm going to talk about the LDL receptor family of receptors. And this is not because the focus is metabolic disease. It's purely a coincidence. I'm not going to talk about cholesterol or dyslipidemia. But the receptor that we happen to be using is part of this family. That's LRP1, one of the longer members. And the receptor was discovered by two different groups working in two extremely different fields. So one group knew that there was an LDL receptor that removed LDL cholesterol from the plasma, but they also knew that chylomicron remnants were not efficiently taken up. So they were looking for the receptor that removes chylomicron remnants. The other group was looking at proteinase degradation. And alpha-2 macroglobulin is a protein that inactivates proteinases and is then taken out of the plasma in a complex with the proteinase. And so these two receptors, the chylomicron remnant and the proteinase alpha-macroglobulin receptor, were both identified separately. And when the molecules were actually sequenced, they were found to be the same thing. And in fact, LRP1 has over 40 natural ligands and is a very high-capacity receptor. This is a schematic of the receptor with a large extracellular domain and an intracellular domain. And this is one of the receptors that's the highest expressed on the blood-brain barrier. There's a little bit of controversy as to where the expression is higher, the apical or the basal side or the luminal side. But the trafficking that LRP1 mediates can go either from plasma to brain or in the other direction, as with some of these ligands. So this is a diagram of the trafficking, and what we do is we've got our peptide conjugated to a drug, and the peptide is recognized by LRP1, which goes into the cell, the capillary endothelial cell, and moves the peptide drug conjugate to the other side. We can do this with a cleavable or a non-cleavable linker. So in some cases, the drug is released and won't be recognized by LRP1. It's a question we get a lot. How do you keep it from going back out by the same mechanism? So with the cleavable linker, it's just not recognized by LRP1 anymore. But there's also something else involved that we probably don't understand entirely, because even with a non-cleavable linker, we still get enhanced permeability into the brain. So the way that we developed the technology was that we looked at the consensus sequences from many of the ligands that bind LRP1, and here are some examples. You can see here is the minimum sequence required to be recognized by LRP1 as a ligand. And we made a library of peptides from 8 to 34 amino acids in length that represented variable sequences around that consensus sequence, and came up with a number of compounds that would go rapidly through a layer of capillary endothelial cells on a dish. So that was our surrogate blood-brain barrier for screening of this peptide. And then we took those through into in vivo models to look at brain transport in animals. So we have a number of these angiopeps. The one I'm going to focus most on is Angiopep 2. The sequence is here. This has been published. We have this patented. This compound, this peptide has been used in many of our programs, and one of them is, I'll mention a little bit later, is in clinical trials, so it's been through tox. So we have the highest comfort level with this particular one. We also found some that don't actually get through, don't bind to LRP1, and those sometimes make nice negative controls. And what I am reminded to mention using this slide is that this is more than a drug delivery technology. A lot of times the impression that people have when they read our papers is that this is kind of like a vector. You mix it together and then your drug gets into the brain. But it's really a lot more complicated than that. We actually do lead optimization programs. So in many cases, it takes numerous constructs, numerous conjugation reactions with various linkers, various different reaction conditions to get various numbers of angiopeps per molecule that we're conjugating. We also can make fusion proteins. And in most of the cases that we've looked at, there isn't really a consistent pattern. So I couldn't tell you, you need 4 of these on here, or you need 2. It really varies from molecule to molecule. And so many of our programs, even though there might just be one slide at the end, represents a considerable amount of discovery and optimization and know-how to get to there. But what we end up with is a new chemical entity that is druggable and crosses the blood-brain barrier, whereas the original molecule may not have. I won't go through this slide in detail, but I just want to mention that our technology has been validated with a number of different kinds of molecules, small molecules, peptides, proteins, and even nanoparticles, which was not work done at Angiochem, but by an academic laboratory at Fudan University. And if I have time, I'll discuss that a little bit as well. So the underlying pattern of how we go about validating the technology is, and making these drug conjugates, is first we do a chemical or fusion connection of the angiopep and the drug. We like to use drugs that have been validated already clinically. Maybe there's a disease that has a peripheral component that's been addressed with a drug, and there's a central component that's not addressed. And so we make these molecules, several rounds of different kinds of molecules, and we'll do an in vitro functional assay to make sure that if it's an enzyme, the activity isn't lost. If it's a receptor agonist or antagonist, the pharmacological properties aren't lost. Then we'll do something which we call the in situ brain perfusion assay. And this is an assay that's done in the live mouse. I'll describe that a little more. And that's to determine both a rate constant for getting into the brain. We can compare conjugate to native molecule. And it's also done to determine the capillary versus parenchyma exposure of the compound. And then, of course, we like to get in vivo a pharmacodynamic assay if there's some acute assay that we can do in animals to give us an idea that we're in the brain at therapeutic levels and able to hit the receptor or enzyme, whatever the target is, and then a full chronic preclinical in vivo assay. So this is just demonstrating the in situ brain perfusion. We give the drug to a mouse in the carotid artery, thereby maximizing the amount of drug that's available to the brain and eliminating sort of liver metabolism, that kind of thing. We'll infuse this for a number of minutes, from 2 to 10 minutes, and take different time points. And using those time points, develop a rate constant. The drug is radio-labeled so that we can measure it from brain at the end of the study. And we also do a capillary depletion method at the end of the study so that we can see what's stuck in capillaries and what's actually in the neural tissue. Just to give you an idea, as you look at some of this K-in data that I may be showing on subsequent slides, glucose has a K-in of almost 10 to the minus 4. Glucose gets into the brain very, very well. It's got a free pass. And alcohol gets into the brain at 2 times 10 to the minus 4. So what we're shooting for when we make our molecules, our peptide drug conjugates, is somewhere in the range of 10 to the minus 3. I'm sorry, glucose is almost 10 to the minus 2. And if you look at some of the compounds that have a lot of difficulty getting into the brain, 10 to the minus 5, 10 to the minus 6 is what you're looking for. So my example of a small molecule is going to be the AN2 paclitaxel conjugate. And this is a relatively simple molecule. It's 3 paclitaxel molecules on 1 AN2. And we have a cleavable linker in this case. The cytotoxicity in vitro is not appreciably modified using the drug versus native paclitaxel. So that's our first box check. And I'm going to skip a bunch of the next ones and go right to the last box check that was done at Angiochem, which is a clinical trial, phase 1/2 trial in glioblastoma and also brain mets. A positive in this trial was defined as complete response, partial response, or stable disease. And in this trial, we had 71% positives in the brain mets and 61% positives in the glioblastoma. These were all patients who had failed prior treatment. In many cases, people had been on more than 10 prior treatments and failed. We didn't see any toxicity related to the platform. The toxicity was the same as what we see with paclitaxel, and we did not see antibody production. And I'm just going to show some images from one particular patient on this trial where there was a tumor. This is a glioblastoma, and you can see the necrotic center of this tumor. Tumor was surgically removed, and you can see the empty space where the tumor had been. Following this, the tumor actually started to come back. It's very difficult to get every single cancer cell, and it's frequent that a tumor will come back after surgery. And tumor was removed again. And unfortunately, only a month later, there's evidence of additional tumor growth. At this point, the individual was put on the trial with our compound, and the tumor was surgically removed again, and it's been 3 years now, and the tumor has not come back. Again, just one individual, but really impressive for that person. We also had the opportunity, because there were so many people in the trial that had surgery for their tumors, we were able to dose 4 hours before the tumor removal surgery and take tumor tissue and measure for drug levels. And what we found was that the drug levels in the tumor were higher than the drug levels in the adjacent normal tissue. And that is probably because tumor cells operate in a hypoxic environment, and hypoxia induces LRP1 expression. So the body has its own natural way of making itself more amenable to treatment for this kind of drug. So that's the small molecule. I'm going to talk about a couple of peptides, first being neurotensin. And this compound, this approach came about because we felt that peptides were very good candidates for the technology. Many of them do not achieve therapeutic levels in the brain when dosed systemically. And there are so many neurotransmitters, neuropeptide neurotransmitters in the brain and neuropeptide receptors that it's a very fertile area. Peptides also have very high specificity. And neurotensin is a tridecapeptide expressed in the CNS, and when administered intrathecally, it has no effects in models of pain in rodents, but when administered centrally, it does induce analgesia. So we thought we would look at this as a model system and see if we could achieve this with our conjugation strategy. So this is what the peptide looks like. You can see the Angiochem on the neurotensin receptor, and the binding site of the neurotensin receptor is circled here. First, the target assay, the pharmacology of the receptor is very similar. Binding to neurotensin 1 and neurotensin 2 receptors is not appreciably altered by conjugation. And this is data from the in situ brain perfusion showing a K-in, native neurotensin. If you look over a period of 4 minutes, will enter the brain at this rate. What we've achieved is an improvement of that rate of about 10-fold in terms of the rate of getting across the brain. And we've done numerous models of pain in mouse and rat with Dr. Philippe Sarret at University of Sherbrooke, and I'm just going to show a couple of them. This is a formalin-induced response where the paw is injected with a nociceptive agent. There's an acute response, which is the pain from having the nociceptive agent introduced. And then there's a longer-term inflammatory response that comes from accumulation of cytokines and other materials to the site. And what you can see is a nice dose-dependent decrease with this compound in the acute response, which is actually something that's not too difficult to achieve with various pain modulators. But more impressive is the response to the inflammatory phase, which tends to be a little bit more refractory to treatment. In addition, we used a model of bone cancer pain because this is a very a highly unmet medical need. And in this model, animals are injected with tumor cells into the femur. And the presence of those tumor cells makes the animal's threshold for pain go down. So if we look 18 days following the injection of the tumor cells at the sham injected animal or the other leg on the other side that wasn't injected, we see that the threshold for pain is maintained, but the leg that was injected, there's a lower threshold for pain as measured by a von Frey system where fibers of varying thickness and force are applied to the foot. And so we see this reduction in the threshold that's noted here. And with treatment, we see this is just different times after treatment. we see a return to almost 50% back to normal compared to the contralateral side. So this is encouraging data that we've actually been able to get this peptide into the brain at therapeutic concentrations, and further validates the technology. The next couple examples I'm going to give are going to be related to obesity diabetes. As Ralph told me that the meeting would be focused on metabolic diseases, I thought I would dig up some older data where compounds were used that pertain to that therapeutic area. I won't go into a lot of background because we've had some great talks earlier that gave excellent background, particularly on GLP-1, which is the first peptide that I'll talk about with metabolic disease. A 39 amino acid receptor agonist for GLP-1 is exendin-4. As you've heard, it enhances glucose-dependent insulin secretion, and GLP-1 receptors are expressed in brain. They're expressed in many regions of the brain, some of which are behind the blood-brain barrier, including the cortex, arcuate nucleus of the hypothalamus, and the thalamic areas. GLP-1 receptors not only alter glucose utilization in the periphery, but they also affect appetite. There is controversy over central treatments for feeding and obesity, particularly with the pulling of rimonabant from the market a few years ago for anhedonic effects. In addition, but contrary to that is these mechanisms do not work by food motivation the way that a CB1 antagonist would. So there has been talk in various circles about even having a brain-penetrant DPP-4 inhibitor. So potentially a GLP-1 agonist that gets into the brain could be a useful therapeutic. If not, it could be a useful research tool to further define the central effects of GLP-1. So in this case, the Angiopep is attached to lysine 39 on the C-terminal of the peptide. And when we look at getting into the brain, this is also in situ brain perfusion, but I'm just showing you one time point with brain and whole brain and the capillary and parenchyma breakdown. And we have a couple of different conjugates which show improved brain penetration and also improved parenchyma penetration, in this case, compared to capillary. So when we looked at OB mice, we used, in addition to control, 2 doses of exendin-4 and 2 doses of AN2 exendin-4, which is Ang 2006. The reason why the doses are different is that this takes into account the weight of the peptide. So these are equivalent doses of exendin-4. Over a relatively short period of time, we saw a reduction in body weight with the Ang 2006 that looks like it may have a slight advantage for the early days over exendin-4 at the same dose, but that advantage goes away as the study continues. So we do not see an improvement in body weight over the native molecule. We do, however, see a little bit of an improvement in insulin secretion over the native molecule, where the low dose of the conjugate gives you better insulin secretion than the low dose of exendin-4. And when we measure plasma glucose, we see that similarly, the low dose of the conjugate is more effective than the low dose of exendin-4. So there are a couple of hypotheses for why we would be getting better efficacy here in terms of the insulin secretion and glycemia, one of which is that maybe we're just getting better exposure to beta cells. But when we look at pancreas, we actually see that the pancreas exposure is the same. The brain exposure is better. This is 15 minutes after an IV treatment. So there may be a central component to it. The other thing I wanted to mention is that the tolerability is better. This is a conditioned taste aversion test. And what it does is it measures the saccharin preference. And if saccharin preference goes down with the drug, it means that there's something aversive in the drug. And Extended 4 shows a dose-dependent decrease in saccharin presence, which is shifted a bit with the peptide conjugate. So that's Extended 4. The next obesity, diabetes-related molecule that we used was leptin. And this is really a proof of concept for a small protein. At this point, this was the biggest thing that had been conjugated in the company, so we didn't know whether this was going to work. Leptin is a 16 kilodalton protein secreted from adipose, and it regulates appetite, metabolism. People become resistant to leptin in obesity, partly because the brain penetrance of leptin is reduced in obese individuals. There's also an alteration of leptin receptor signaling in obesity. So this is just a list of some of the fusion proteins that we made with the leptin molecule. We used fixed linkers, flexible linkers, C-terminus, N-terminus. And in the conjugate, in the fusion protein that we chose, actually none of the fusion proteins affected the binding. The KD is the same to human leptin. And in a competition binding assay, we also see similar affinity. The same is true for leptin signaling. So p-ERK and p-STAT were measured here. These are downstream proteins, phosphoproteins, that participate in the leptin pathway, and we didn't see changes there. So if we do the in situ brain perfusion assay, you can see that leptin actually does get into the brain, which is good. It's supposed to. It's secreted by adipose, and it has central effects. But we can improve this by about 5-fold. And in a study in DIO mice, this was a longer study, we can see that the animals gain weight, and even the leptin-treated animals in this study gain weight, but the two different fusion proteins showed either weight loss or, on this day, a reduction in weight gain. So this was the first in vivo evidence that the company had that proteins could actually be, that protein transport across the blood-brain barrier could actually be enabled with this technology. So I'm going to switch gears a little bit and talk about some data that was generated at Fudan University using nanoparticles. Because one of the things that many individuals are interested in is using siRNA or using gene therapy, and nanoparticles seem to be a pretty common method for doing this, at least in vitro. So what they did was they took these nanoparticles and attached the AN2 to the nanoparticles, and you can associate DNA with that. And what they did was they used GFP, DNA that encoded GFP. And when they injected the uncoded nanoparticles, there's not much in brain. You can see it goes to liver. With the Angiochem-coded nanoparticles, we do have brain exposure that you can see the fluorescence. And if you look at the various, at the microscopic level, various brain regions, you can see that the DNA is actually producing GFP in these cells. So that could be a further application of the technology. At this point, we've also worked with monoclonal antibodies. And gotten them into the brain. We've worked with enzymes. We've started some programs on lysosomal storage disease enzymes. And these enzymes are mostly these are rare diseases. But the technology or the enzyme replacement therapy has been validated clinically. Patients are actually improving and they're living beyond childhood with this therapy, but the central symptoms of the lysosomal storage diseases are not met. So we're hoping to use this technology to make that enzyme replacement therapy effective across the whole body and really make an impact on that disease. So I'm going to stop there and acknowledge the folks in the company who have done all this work. I particularly want to acknowledge Michel Demeaux, who is our scientific founder and directs the operations in the lab. He has been with it since the beginning and has acquired a wealth of information and experience with this technology. And we do have our collaborators that I want to mention. Steve Gonius at UCSD is an LRP1 specialist. Caroline Nikolic. Frank Pareca helped us out on the pain program. Philippe Serret also on the pain program. And Quentin Smith is an expert on the blood-brain barrier and actually developed the in situ brain perfusion and K-in methodology. So thank you very much.
So can you tell us when these things get to the site of action in the brain, how long do they stick around?
Yeah, so we have done PK studies on some of our conjugates, and the longest we've looked is 72 hours because for the antibodies, we want to dose twice weekly. That's typical for in vivo studies for a lot of these MABs. And we do see there's therapeutic concentration in there. And when I say therapeutic concentration, I mean 3 times the in vitro concentration needed to be cytotoxic at 72 hours.
So the other question is, where else do these conjugates go?
Yeah, so we are not exclusively hitting brain. LRP1 receptor is expressed on lots of different cells. Liver, bone, it's everywhere. So this is why in enzyme replacement therapy, for example, this will be the drug that you take. You don't need to take this plus a peripheral drug. So we're not targeting brain at the expense of other tissues. It'll go everywhere. In many cases, we see the same PK, oddly enough, as the native molecule. But in some cases, we do see a reduction in plasma AUC, probably because the LRP1 in the liver is grabbing all the molecule. But I'm not sure why, but it's compound dependent.
We have time for more questions if anyone has them.
You said that it's a very high capacity receptor and turns over quickly. But is there a maximum threshold of how much can actually be transported or no? Have you reached the maximum level?
Yeah, so we can hit it, but we have to give very high concentrations, like millimolar concentrations of our peptide in order to block, if we're using RAP, which is an endogenous ligand, we have to go very high. And I think that's part of the reason why this technology is working well where others have failed. So that and the low affinity. we're not looking for high affinity interaction here. We want to get dumped off that receptor once we get in. So if you look at, there have been attempts using transferrin receptor, insulin receptor. These are lower capacity. They don't have numerous ligands, so the expression doesn't have to be as high. And there are also many of the strategies have involved high affinity, so the molecule sticks to that receptor and doesn't actually get out into the brain where it needs to be.
So I guess a follow-up to that, but what is the affinity and the off rate for the peptide for the receptor?
So the affinity of the peptide for the receptor is in the hundreds of nanomolar. And in fact, there are times when we conjugate and we're in the micromolar range. So it's not necessary to have nanomolar affinity. We haven't done extensive kinetics, so I can't tell you the off rate specifically, but the whole process takes about 30 seconds to get from one side of the cell to the other.
I noticed on the small molecule Taxol that you actually conjugated through some of the lysines of the
Yep.
peptide. Does that affect its binding to the receptor and how it gets in? And also, when you do that to affect solubility, peptide seems fairly hydrophobic, so I don't know if you have solubility issues that come up with some conjugates.
Yeah, so we have had solubility issues. I'll answer your second one first. We do typically have to work with formulating and things like that, but we've also had molecules that were fairly soluble. It's not an insurmountable problem. Usually salt form selection is enough to fix it. The lysine conjugation doesn't seem to affect the binding. At least we haven't seen it do so at this point.
What was your solubility for the peptide with 3 paclitaxels on it? Do you know?
I don't know the exact number for solubility, but it's given as an IV infusion, and so it's acceptable for that formulation.
Are high levels of chylomicrons competing? So this is an administration that should be done in the fasted state, not fed state?
We haven't actually done fasted-fed comparisons. In most cases in our clinical trial, these patients are in the fasted state when they come to the clinic just because they're accustomed to receiving agents that are going to be emetic. So we have only looked in the clinic at fasted state, and generally in our animal experiments, we're injecting fasted animals too. But that's a great question. That's something that could make a difference.
Okay, if there's no further questions, let's thank Gene again.